First of all, thank you very much for this invitation to share some of the work that we do in our lab. I recently joined the ELIXIR and the systems biology group. I'm still learning to know the names, the faces, and what the activities are. But we met in the last all hands meeting, and I'm on the discussions. Well, the idea came up of presenting some of the work that we are doing related to systems biology. I am a research professor at the Institute for Integrative Systems Biology of the Spanish National Research Council. I'm also a courtesy professor at the University of Florida, and we are located in Valencia, Spain. My lab is mostly devoted to the development of methods for transcriptomics, multi-omics data integration. So we have been working for quite some time on how to integrate and how to analyze multi-omics data altogether. And I wanted to share with you some of the ideas or the worries that I still have on how we are dealing with the different signal-to-noise ratios that we find in these types of data, but also the annotation of the experiments, of the multi-omics experiments by researchers. So to give some context, we some time ago, a couple of years ago, we published this perspective paper in Nature Computational Sciences, where we were kind of offering our view of how we are using multi-omics data and what the challenges are. And basically, the way we see this is that, of course, all these omics layers, I don't have to explain, but basically the way we are using this information is like two ways of two possible manners of using the information. Sometimes what you have is that your interest is mainly focused on the samples and you use the multi-omics information to cluster, for example, different disease groups, and even to predict when you have a new patient in which of the disease groups will correspond or what will happen with this patient in the future. So here is kind of you use this information as biomarkers and then trying to predict or to focus on the samples. But in some other cases, it's the other way around. You more focus on the features, so you focus on the genes or the metabolites of the proteins, and you hope that by putting all this information together, you somehow can infer these multi-layer networks and then see how they change from different conditions. And of course, here in this context, maybe this concept of biomarkers will be more related to which are the hubs or which are good elements of key elements. So, different analysis approaches are trying to, to go one way or another, but also in the way in which we are storing and annotating and handling the information is very different whether you want to be here or you want to be there. So my lab is more interested in this second part in which we are really trying to understand the biology from the multi-omics data that just predicts or just that predict. So we also highlighted a number of things that we saw that were important. They were the challenges that you probably recognize. We have non-uniform missing data in many of these data sets. So the number of missings is not the same across different omics modalities. The problems with computations, I don't have to mention. The differences between in the signal-to-noise ratio across the different technologies is what I will be talking a moment about. I think still we have trouble to interpreting comprehensively this information. Of course, we may have also some problems of some risk related to when we try to put all the data together and do meta-analysis and not taking into account some biases that may be part of the data. And then some other things are related to the annotation, and I will also talk about this. So basically today I will talk about these two things. As I mentioned before, my lab has been working in a lot of different tools. So we are mostly developing methods and tools in which we are trying to go for these multi-omics data sets to understanding something on the biology. And there we have things about experimental designs, the quality control, the storage, statistical analysis, and visualization. And today, as I mentioned, we will talk about power analysis that is very much related to these signal-to-noise ratio differences. And this development is the Stategra EMS, that is a tool that we developed trying to address the problem of storage, of annotation, I would say. These two developments are published already, and you can find the first part of the talk will be about this power analysis published in Nature Communications, and the Stategra EMS was published in BNC Synthesis Biology. So for some of the data that I'm going to present, it has been of some of the methods. They have been developed on these multi-omics data sets that we generated in the context of the Stategra project. That was a project with EU project in which we were trying to develop methodologies for multi-omics data analysis. And what the data set I want to explain you, because it was very relevant for the type of research that we wanted to do here, was about, in this case, B-cell differentiation data sets in mouse, in which we were starting from a particular cell line, the B-cell cell line, which is part of the lineage, and is a cell line that is in the cycling pre-B stage. And what happens is that in this cell line, when we trigger the expression of the Icarus transcription factor, this cell line starts cycling and start differentiating and proliferating, right? And then it can go to the next stage in the B-cell differentiation. So what we have here was these experiments in which we were starting the change in of the development, the differentiation of this cell line, and then we will be taking samples at different time points for 24 hours up to 6 time points. And then, of course, we have our controls, and then we measured all these types of omics. We had, of course, transcriptomics, but also microRNA, reduced representation by sulfide sequencing, which is the DNA methylation, chromatin accessibility with DNA-seq, proteomics, metabolomics. We also have ChIP-seq, single-cell RNA-seq, and so forth. So it was a large data set. So the good thing of having this in vitro system is that we could generate enough amount of cells to be able to do all these assays, which is something that normally does not happen. Sorry. So, when you get the data, then you realize a number of things. And probably you all know about this, but I think it's very nice to put them and to see them in context. So what we found was that all these omics that they are generated in the same system, in the same samples, actually have very different dimensions. So, for example, the DNA methylation data had 1 million of features, while the metabolomics data was much more smaller, and the RNA-seq was some half of the intermediate. So we don't have the equivalent, not even equivalent, number of features in each of the omics data set. Because of that, in relation to that, we also have different coverage of the targeted space. And what do I mean by that? So, for example, if you are trying to measure gene expression, well, there are a number of transcripts that are expressed. So, are you capturing them all in your experiment? So what you have here is a representation of this density plot of the genes that were captured with RNA-seq. So we have like some, mean level of expression. This will be the low expressed genes, and this will be the highly expressed gene. So if we now take all these others technologies and ask, how are all these genes represented by the other omics layers, at least those that can be related to genes. So what we find is that, for example, those related to chromatin structure, like DNA methylation of ChIP-seq, kind of recapitulate most of this this space, but it's clearly that they have less power to capture those genes that were lowly expressed. If we look, for example, to single-cell RNA-seq, then it's even more clear than that, right? So everything that is low expressed will not be captured. So we are only looking here with this technology. And when we look at proteomics, this is even stronger. So actually, the picture that we get, for example, from proteomics is just a fraction, at least a fraction. There will be also other differences, but at least a fraction of the picture that we can get with transcriptomics. So that means that when we are trying to integrate the data and, for example, develop this correlation of networks, we really have to understand that we don't have the same coverage of the space, of the targeted space with the different technologies. So we are not seeing with the same intensity. The other thing that happens, as I mentioned, is that we have different noise levels. And here you have a representation for this particular data set. It's not the same for every data set, but the fact that we have differences is consistent, in which we are representing the signal chain. So it's like the dynamic range of the signal of the technology, if you wish, that we have for the different omics versus the variability per condition that will be like the noise, right? So this is the range of the signal versus the noise. And you can see that, again, that this is very different. In the center, we have, again, RNA-seq that has quite a decent range of signal and moderate noise. But if we go, for example, here in the case of methylation, the noise is much higher, at least in our experiment. And if we go, for example, here, that was the ChIP-seq, it was very different of in the proteomics, we also had a much higher error. So again, the capacity of each technology to detect changes is different because the levels are different. And also we have, as I mentioned before, different dynamic ranges. And this is another type of experiment, but in which we are comparing log-fold changes. Here on the left is on gene expression, and this is ChIP-seq in two different conditions. But I want that you see that the the dynamic range of gene expression and in this ChIP-seq was different. But actually, these dots that you see here correspond to the same gene. So actually, even if you have these differences in dynamic range, in this case, it's like the order of the feature of the relative proportions may be consistent. But again, the capacity of one technology and another to capture the signal is different. So with that, what we did was to define what we call the figured merits, what is called in analytical chemistry, the figured merits, that somehow is something used to describe a technology. And basically is a way to see how the true signal relates to the measured value, right? So you have, of course, sensitivity, so it's your capacity to detect. So with the measure value to detect what is a true signal, you have some linear range in which this relationship is kind of linear. You have the dynamic range in which you really, is the amount of signal that you can capture. And then you have things like the reproducibility that will be related to how much dispersion you have in your measurements. So we did some exercises, and I mean, it's a long discussion in the paper. I really invite you to look at that. So we did some exercises in trying to understand how these figured merits are actually different across omics layers, right? For example, what is critical for sensitivity in proteomics and metabolomics? But here, of course, it's very much depending on the machine, right? So, and the technique that you use is determining how your sensitivity is. Maybe in more like sequencing-based approaches, sensitivity is much dependent on the sequencing depth, for example, because features that with a lot of reads will be detected better. And also in the case of region-based approaches, we also have some other type of considerations regarding sensitivity. If you can see, this is always, I mean, there is always something different. If we also look to reproducibility, but usually those features that are more highly abundant are more reproducible, but also it's important what is the error of the technology, the limit of the detection will also have different consideration in its methods, and then we made kind of the exercise of trying to describe this for each of the figures merits. Okay, so I will not go more in details on that. So how we are then using this in terms of thinking of how we do a proper experimental design in the situation of a multi-omics experiment. So what we understand here is that when you are performing, one of these experiments, you usually, if you think only on one feature that you are measuring, you have a number of observations, for example, in two conditions, right? And there will be some effect size that somebody wants to detect, and also some dispersion of the data, as we mentioned before, right? But in the context of multi-omics, we also have many features. So that means that we have also to take into account, for example, multiple testing correction because of this large amount of features. And then in the case of omics, then when we have several omics, then we have also the other dimension in which, as we mentioned before, right? So the number of features will be typically different. So the kind of adjustment that you have to do is different. So with that, we developed this multi-power tool that basically what we do is to take a number of parameters from a data set, from existing data sets. Usually, if you have some pilot data is good. So we can use what we call the cohesivity to calculate these effect size, but also which is the false discovery rate that you are wanting to have in your experiment, which will be the typical number of differential expressions of differentially abundant features, and which is the desired power that you will like to have in general or per omic. We consider whether the data has different distribution, because this is also something that is different. Different omics data has also follow different distributions. And then we perform an optimization problem in which we are trying to identify which is, the number of samples per omics and per condition that we will require, also taking into account the cost of the technologies, and in order to have a specific power globally or in each case. So the result of that is a graph like this, in which you have, for the particular experiment that you are looking at, you have a graph that represents the sample size that you require versus the statistical power. And for example, what you can see here is that if you want to achieve the same statistical power in two very different omics, for example, this will be RNA-seq and proteomics, the number of samples, the number of the replication that you obtain, that you require, is very different. But you can also go the other way, so you can establish, the number of samples that you may are able to perform, and then understand how the power will be different in one technology and the other. In with this information, design your experiment. You can also say, okay, so I'm going to be measuring less or more features, so that my multiple texting correction problem changes, and in this way you can somehow adapt what is the power that you will obtain. And I will stop it here for this part of the presentation. If you want more details on this, you can read the paper. Oh, sorry. No, I forgot this one. So when we submitted the paper, we were also asked to somehow provide a way to calculate power in other type of experiments in which we are actually doing prediction. And it was a kind of tricky because actually there were not methods of established methods in which how you calculate sample size in terms of predictive power in the context of a multi-omics experiment. So we came up with this idea in which we'll basically what we did was start with our experimental data sets and with an increasing number of samples, calculate the classification error, and then extrapolate and what will be the for a desired, let's say, prediction accuracy, the number of samples that we will require. And then we can do this in this, this is another piece of software, multi-ML. You can do this for different omics modalities, or you can put them all together. So at the end of the day, you will, you can actually calculate which are the number of samples that you need in each of the omics for reaching a particular predictive error. And again, if you put the cost on top of that, you can calculate which is the most cost-effective approach. And this is also available for you. And then I wanted to say a few words about this other part, which is about annotation of multi-omics data sets. That was also developed in the context of the Stategra projects. And basically what we had there was the fact that we were generating this big data set with all these samples, different labs were involved, different instruments, different batches, and we really did not find a good way to capture all this information together. There are some other things that are out there, for example, omics. The eye is collecting and annotating data sets from the community, but it's not really focused on the experiment of one lab, let's say. And projects like, for example, ENCODE, they have a very nice way to annotate all the data, but it's proprietary. Obviously, something that they use, and it's not easy to extrapolate to other settings. So we wanted to have something that was really for the people, right? So that's for experimentalists to annotate the data. So we came up with this Stategra EMS, which is a management system that's kind of works as a logbook to be able to annotate your multi-omics experiments, and also the measurements and the analysis pipelines. So basically, the way we did that was to organize the data around 3 modules. So the biggest module is the experiment module, in which you have the experimental design information. So you describe your experiment. The sample usually will be the used biomaterial, and the analysis pipelines and results. It will be how you analyze the data and how you are obtaining your process data. And what is important in the conceptualization is that in the sample module, we thought of, okay, so what is the biological conditions, the biological replicate, and the analytical sample, because they are kind of different things. And of course, in analysis, you have the raw, intermediate, and process data. So this is like an example of what could be in terms of, the complexity of the experimental designs that we were trying to be able to accommodate. So imagine that you have a particular cell line, a biological condition, and then you have a number of this particular biological condition. You have a number of biological replicates that are the actual, let's say, like sample. You may have different batches, and then you may have that each of these replicate is used to for one assay, right? So for example, for RNA-seq, for chip-seq, and then you may have situation in which a biological replicate, one of the biological replicate is for use for one assay, but you can also have the situation in which the very, very same sample is used for different extractions and then from different measurements. So and this is different. So the way in which we design this database is able to capture this type of complexity of relationship between these three elements. Basically, we have an experiment form in which you describe the experiment, experimental design, you say, which are the components, there is a summary, then you can go by the biomaterial, the experimental condition, and then start annotating all this information. And then with this kind of graph, once you have defined what is the information that is in experimental condition, biological sample, and replicate, analyze, replicate, and you can connect them in this way so that everything can be traced back. And then we have all the part of that is related more for the analysis, and again, so you will have that from your experiment. You first obtain raw data that typically comes from each analytical sample, and then you may have some kind of intermediate analysis in which you may do some pre-processing or mapping or data smoothing or normalization, and then you can go further into what will be like the final true measurement that you can say that this is the measurement that I actually have from my original data. And this is again all connected back to the origin of the information. And this is again how you define this. You can click each of the steps that you want to record, in which you explain how this step starts from the previous one or the previous sample, and you can have this overview. And when you click and create a new analysis steps, then there will be another window in which you can create add all the information, whether this is the raw data or this is how you did the analysis. Usually this is just for annotation, so we don't really envision this as a way to store the data. So if you have, for example, a FASTQ file, you just have the file location, right? But not the actual data is not in the database. And then we implemented, standards so that you can use, a little bit the community standards to annotate all these elements. And with that, I don't know how much time I used, but these are the two developments I wanted to share with you. Most of the multi-omics work on statistical work was done by Sonia Tarazona and Leandro Balzano, and Rafael was involved in the Stategra EMS. Of course, the team was very important to provide all the data in the discussion, and then just acknowledging the funding sources of these projects.